there is some suggestion with when we did give it in animal models in later disease stages that we did see, you know, less scarring and things like that than what they started with. And so I'm not going to say it's a cure, but I think it has potential to even potentially delay late disease remodeling.
Do you have a cardiomyopathy and are hoping for a way to stop the progression of your disease or even reverse it? Today's guest on Let's Talk SADS Live, Dr. Farah Sheikh, may have that solution. Dr. Sheikh is a renowned Canadian scientist and researcher working as a professor of medicine at the University of California, San Diego. She and her colleagues developed gene therapy to modify rare genetic cardiac conditions like ARVC, and their company was acquired by Lexeo Therapeutics in 2021. Lexeo is now running a clinical trial to add fully functional PKP2 cardiac muscles to ARVC patients. This could stop or even reverse the progression of ARVC, and it's possible that this gene therapy will work on a multitude of rare cardiac diseases. If you're curious about how gene therapy could shape the future for people with SADS conditions, this episode will help you better understand what it will mean for you and your family. Hi, and welcome to Let's Talk SADS Live. Today, my guest is Dr. Sheikh. She is joining us from sunny San Diego. We are very jealous as I'm sitting here in cold Toronto. Dr. Sheikh, you are a consultant now with Lexeo, which is running a gene therapy trial for ARVC. This is really exciting news, and we're so pleased that you, as the originator of the research with your partner, are here to talk to us. We understand this is also your first podcast, so we're very, very excited to have you here. So before we begin and get into the research, can you tell us a little bit about yourself, your background, your training, and also you have a personal connection, I understand, with ARVC, so maybe you could share that with us.
Sure. And I want to thank you so much for inviting me. I am so excited to be here. As I said, as you also said, it's my first podcast. Happy to share about my journey with ARVC. So, just to give you a little bit of background, I am Canadian. I was born in Chibougamau, Quebec, which is very up north in Quebec. And to be honest, my journey with the heart really began because my father suffered a heart attack very early on in his life. He was actually, I believe, in his late 30s, so very young. And, you know, that really shaped my career because I always wanted to find something that could be helpful for him. Unfortunately, he has since passed of a heart attack, but it just struck me that in your life, sometimes you often find yourself in a similar position, but different, maybe disease front. And so my whole training has been really done in Canada. And so that's where I did my undergraduate at McMaster University. I then went on to do my PhD at University of Manitoba. And then I came to San Diego to try the opposite extreme in terms of weather, to sort of fortify my training, working with one of the leading groups in molecular cardiology, because they were very interested in genetically engineered animal models to better emulate human disease so that we could understand disease a lot better. And as I was going through my training and I had an opportunity to move up the ladder, as they call it, I started forging my own sort of career. And, my whole research and career has been about looking at the heart muscle. And I really want to understand the architecture of that. And so I was studying other parts of the architecture of the muscle when I got to what I call is the cell-cell junction. And so as I was trying to understand the cell-cell junction, and this was sort of my own independent career, unfortunately, someone in my life got diagnosed with ARVC. And it was such a weird coincidence because at that time, there were no models for animal models that really emulated the disease very well. And I was trying to develop such a model. And then this person in my life got diagnosed. And I was like, wow, this is such a coincidence that, and sometimes you often get signs from above, from wherever, to direct you in your own sort of where research, where you should go in your life and path. And I really thought this was a sign that I really had to dig deeper into that. And I also understood, and as we know, unless you do something about it, no one else will do something about it. And so that was sort of my fortitude into moving into this area. And first of all, just generating animal models that better emulate the human disease. And once I sort of did that work, then I started to understand the molecules that might be driving this and what is the order in which these molecules fall apart, unfortunately, at that location, and how can I intervene? And so that's where I started developing approaches where I sort of bring back the healthy gene, or what we now call as gene therapy, as a way to intervene with the disease. So that's sort of the sequela of events that led to my research career.
Wow. It's amazing that you were already researching it and then someone in your life was
Yeah.
diagnosed, not the other way around, which is sort of
No.
a classic researcher story, I think. For our listeners that are newly diagnosed, can you just give us the sort of quick and dirty, what is ARVC? What are the markers of it? A little bit of the background of what that disease is and what are the outcomes of it?
Sure. So I think when we think about, in trying to look at these models from early to late and how they correlate with human disease, a lot of times and that's why it's called arrhythmogenic, is the arrhythmias come out first. And so one of the first signs is a lot of these heart rhythm problems, which with exercise, because we know that somehow adrenaline fuels the arrhythmias, that's sort of the first signs of the disease, and often with exercise. So not necessarily sitting on a chair and looking at your heartbeat, but actually with exercise or stress, that's when you get arrhythmias. And we see that also in our animal models, like without any structural problem to the heart, these things are sort of happening. And then I think there's this very important transition that happens where, there's the falling apart of these junctions. So, there's various sort of structures there. And one of them is the electrical communicator. And this is why we get arrhythmias and the electrical communication is gone. But then you get the mechanical connectors, which is the desmosomes. And once you start to chip away at those proteins, then, you get sort of muscle death, muscle loss, scarring that will occur. And then instead of, as I said, when muscle death occurs, muscles can't divide. So that's why the scar comes in. And what's unique for ARVC is sometimes fat comes in. And so these fat cells are also populating this. And so you could imagine the muscle cells are the ones that take up and all the nutrients, the oxygen, and sort of are the pumping and allow you to pump strongly. But when you lose those muscle cells, that's where you sort of get all these other cell types that don't have that same capacity. And as a result, it's a stiffer heart because of all of these other cell types that are there. So that will lead to loss of muscle function. And so oftentimes, which we still don't understand why the right ventricle is primarily affected in some patients or first affected, but that is something that gets to, you lose function of the right ventricle and eventually the left ventricle. And oftentimes at the end, it may, or, in a more end stage part, you will get something looking like heart failure because, you just aren't able to sustain what's coming into the heart and getting it out of the heart in a very efficient manner.
When you first started, what did you define as the problem that you wanted to solve in your research?
So the first thing is the animal models. I think, I think I wanted to understand the disease at a very early time point. And with often looking at patients, you kind of get a snapshot, but you don't get the necessarily the evolution of what happens with the disease. And so with animal models, you can actually look at that. And now we are using stem cells. So you can actually take skin or other cells like blood and turn them into sort of stem cells that you can then push to become muscle cells. And you can look at the very, very early nature of what happens in those human heart muscle cells from that system. But I think it's important to understand what are the early drivers, because that's also where you're going to get to the root cause of why things are going wrong. And oftentimes that's why gene therapy is a great modality, because it attacks the root cause, the gene where the mistake has happened, that you're now trying to replace that healthy gene.
And you're looking specifically at the protein PKP2. And when I was reading, that's correct, right? In this
Um, so
instance?
that is one of the genes, but I do look at other genes, but
Okay.
plakophilin 2 is one of the most, commonly mutated genes in that population. So definitely that's one that we look at, but we also look at others.
Okay.
Yeah, desmoplakin, for example, I'm gonna just put out there.
So, is it correct to say that the genes that you're looking at are mutated or not enough of them?
So this is exactly what we wanted to find out. Is it that the genes are mutated and causing the protein, which is the outcome from the gene, to form something toxic to the cell? Or is it that you just get less of it there? That's the normal form,
Mm.
because it's now converted into something different. And so that was one of the premier kind of models that we really focused on, is trying to make a model which had the human genetics and had one of the number one mutations found in that population, and found that, you certainly get the mistake, you certainly get a gene, and also a protein that looks different, but you get so much less of it. So whatever's there is probably not as stable. But we did an experiment in cells really to show that when you boost that mutant form, you can actually get the cell to do better. So it's just that not enough of it is there. And even if you give the normal healthy gene, you can also boost everything. So it's about the quantity. It's not about the quality of that mutation. In fact, we think from our research is that the mutation actually allows you to survive. It gives you some benefit to go to birth. And as a result, because it's still hangs on to a lot of those mechanical anchoring actions of that junction. But, over time, because once you're born, now you're under your own circulation, you have a lot more pressure on your heart, and it may not allow you to thrive per se. And especially, of course, in exercise conditions where you need the heart muscle to be very, very intact, that may be more, it might be more difficult in that state.
So this is fascinating for me to hear this as someone who was diagnosed at age 21. I'm 56 now, and I'd always heard it was a mutation, and I never had regarded it as actually I didn't have enough of it, but the ones I had, and if you boost those mutated ones, that could actually be pretty good for me.
Yes.
Like that's a big
So,
breakthrough.
yeah, so I think that is what the novelty of a lot of the work that we did. And because, also people thought that essentially maybe one gene is not enough because the whole architecture is falling apart. And so that was the other important component, that one gene may be enough to change everything. And the mutant form actually is helping you in some ways. And just having even more of that be more stable, for instance, might actually prolong, the way the heart performs in a good way.
Wow. I am going to be much more thankful of my mutated PKP2 gene now. I'd be so much kinder. I'll be angry at it. So tell me about how you have basically prioritized the development of strategies to boost production of those genes.
So, what we found in our research, and I sort of alluded to a little bit, is that when we give back just the healthy copy, I mean, ideally, we would want to give back the healthy copy. And that in and of itself could bring all the junctional proteins back to alignment. Let's put it that way. And this had a profound effect on preventing muscle death because we saw less scarring, less of all of those things that you want, or that you don't want happening over time. And then it boosted the function of the heart. And in our animal model, there was a lot of lethality, and it could prolong life of those animals. They, 100% survival. And when we gave it in disease states, so of course mice and human are different, but we gave them at a time point in the mouse's lifetime where they were diseased, and we saw significant benefit even in that setting. So I think we use gene therapy as an approach where we drive higher levels. We put some sort of controller in front of the gene, or a promoter as we call it, and only targeting to heart muscle. So it doesn't go anywhere else. It's only going to be localized to heart muscle, and we produce more because when the gene goes in, well, then it will form the message, which will form the protein, and then go to those junctions and sort of bring them back together, sort of stitch that component back together there.
So let's just like dumb it down, because pretend I'm a Martian. I just arrived on Earth and I don't know what you're talking about. So if I was deemed eligible to participate in the LX 2020 heroic PKP2 clinical trials that Lexio is conducting right now, what would I be given? How is it given? Like, just walk through just a very basic stuff. Like, is it a shot? Is it a pill? Is it something in my ear? Like what is it?
So basically, we just give a one-time injection. And so this is, and so I'm going to preface myself that I'm not a clinician. And so I'm not directly doing this because, I'm involved in sort of the non-clinical sort of, and I, of course, inform everyone about what we see in animal models and how it can relate to people, but I'm not giving this. But as far as I know, they give it similarly, where they do it a one-time infusion injection, and that's pretty much it. You go in there, you of course have to meet certain eligibility criteria, which they have put together. And as again, I'm not a clinical person, but clearly they want to see some disease from the patients having this PKP2 mutation. So that's definitely something that is there. And but we want to be able to give this one-time injection and follow the course of your your lifetime with this particular injection. So it's a one-time shot, in other words.
And what would success look like? What is it not, and what can it do, and what can't it do?
Right. So it is not a cure. I would not preface it as a cure, because a cure, I think, would be almost like you would never get any sign of the disease. And so we have to be very cognizant of how we put that messaging out, because when we, of course, even give it to mice, there is some disease, but it affects function and all of those outcomes, but you may still have some part of that disease. And so the idea is that you are changing the way the disease behaves in you. And so it will change. Hopefully, my hope is, and I think Lexio has already reported early on in March some data that it changed PVC burden in patients. So I think that's the number one thing that we also see as the early sign of the disease and things like that. And hope with later time points that they'll see impact on, remodeling of the heart and not having this dysfunction and things like that. So I think I want to just preface it is it's going to change the way your disease you may be operating in the sense of the disease in a very mild setting versus going towards a very severe setting or heart failure.
So is it fair to say, Dr. Sheikh, that it stops the progression or the advancement of the disease, or is that correct to say that?
It's correct to say that. Now, we do have some data that suggests that there is potential reversibility, and those are in our publications. But that is something that we would, we don't know if that part truly translates to people or not. But I'm hopeful, but I can't say that right now. And so there is some suggestion with when we did give it in animal models in later disease stages that we did see, less scarring and things like that than what they started with. And so I'm not going to say it's a cure, but I think it has potential to even potentially delay late disease remodeling.
That's incredible. I mean, I can't listen to this without thinking about myself, because for the past 11 years I've been living with heart failure as a progression of ARVC. To imagine that patients like myself and others can reverse some of the scarring and have increased heart capacity and function, even still having the diseases, because he says it's not a cure. I mean, that would really improve quality of life.
Right. And I think that's what I am always mindful of. Like, even that is the ability to exercise is probably number one question, I'm sure, for everyone.
Yeah. I'd love to play tennis again.
But, yeah. So, I mean, I think, we have been closer, like minus 5 years ago. We're a lot closer than 5 years ago to having something that may have a therapeutic transformation for patients in some ways, and I think going after the root cause is partly due to that. We cannot sort of completely know until we do the clinical trials that they're, safety first, of course. And that's how, thinking about Lexio and how we've interacted together, they are always mindful of safety first. And
sure.
So I think, we want to just be cautiously optimistic. I guess that's the point. You should always have, as a researcher, I'm always critical of every step I take, and you have to have that same critical nature whenever you're going to go into something like a clinical trial. So
absolutely. And we should preface this that if people are considering this is obviously a conversation they have with their clinicians and then with their families to understand the risks. We're not giving advice on this podcast whether people should do it or not do it. Go and speak to your clinician. I have a question about, this is an inherited disease. This runs in families. Is there an anticipation on how early someone might be able to take this? If a child is born, it's known as they inherited the genetic mutation. Could this, if administered early, could it just not, the genes or the disease just doesn't get expressed?
So that's what we found with when we gave the intervention or the gene therapy very early on in our mice, like almost like when they were born. That definitely changed the scenario for that animal model to look like it did not have any level of disease. But I think, we still have to understand what is early and also there's a lot of conversations that have to be had with your cardiologist about the type of mutation you have. I mean, variants of unknown significance or or those that have been shown to have pathogenicity. So I think there's a lot of conversations that have to be had related to that. And, I'm sure, yeah, it opens up a whole area. I wish, to be honest, it could be a newborn screen and be used as a way to, at least, because we know even with patients now, right? Like, you were diagnosed at 21 and you've been able to live with the disease, but that's through a lot of care, and monitoring and all of those sorts of things. So we know, I think, just with patient care, the earlier you intervene, the better off you are in terms of your disease, at least for ARVC. So I think, I would love that, but I don't know, I'm not the clinical person that's going to be, um, uh, potentially figuring out all the nuances that relate to sort of putting something like that together. But I would love to be a part of the effort in advocating for that.
Great. Can I ask you more broadly, this approach that you've taken, well, is it disease as agnostic? Like, could this work on other cardiac conditions in our SADS group, or which is like long QT? Is your approach able to move across other cardiac diseases, do you think?
So we're looking at that now because, this is, even with ARVC, there's multiple mutations, right? And to be honest, I am looking for mutation agnostic therapeutics. I think it should be given to all and not just, but I think the PKP2 is sort of maybe leads the way because there's just such a patient population, as well as a straight gene replacement approach per se,
Mm-hmm.
that is sort of testing the ground, for these types of potentially mutation agnostic therapies. And so I think, you always have to go things stepwise. But definitely, I do have mutation agnostic therapeutics that I am working on right now, and that also Lexio has within their asset portfolio that could, be important for patients with DSP cardiomyopathy, which is very hard to target, because the gene is just so large. And so you may not be able to package it in a way that's efficient to then deliver the healthy copy. But I think, I want it accessible to all patients. Like that was honestly my goal, because of course, PKP2 is something that, just came on my lap in a way because of all of the conditions that surrounded all of that. But I really want to go for a mutation agnostic therapeutic so that patients can equally benefit, even patients that don't have a mutation, because maybe there is some value to having fortifying those connections.
Absolutely. I mean, my sister is gene elusive. So even within a family where someone's diagnosed with PKP2, another family member could have no known cause.
Right.
But maybe
Absolutely.
we haven't discovered that gene mutation yet. Maybe I have a double dose, but if the reducing the burden of the disease can come from a disease agnostic approach,
Absolutely.
the quality of life could improve, the quantity of life could improve. This is really groundbreaking stuff. This is the last question. What do you see on the horizon? Like, what is getting you out of bed now? What are you working on? What do you see coming down the pipe that maybe we're not aware of?
So I think what you're going to see is that people, of course, the cardiomyocyte is the important muscle, the cell type in the heart that everyone is focused on. But I think there are a lot of supporting characters to that heart muscle that we don't understand the crosstalk of what may drive severity of disease. Because maybe, it's those supporting characters that are supporting the cardiac muscle that may drive the severity of why one patient may have worse, or and so it could be exposures to inflammation, or so inflammation is like a huge area that I think, quite a few people in the field are focusing on, because that may be the differentiator as to why one person is more severe, less severe potentially. And so I think I want to understand, well, what's the first hit? What can drive, everyone may have these mutations, and we also see families with the same mutations with different severity of diseases. And you're like, there's something there that we need to dissect better. And could it be the supporting cast? In other words, what other cells are interacting with the cardiac muscle cell to drive sort of the disease burden and things like that. So I'm very much excited about that area and trying to find something that could be the trigger, and trying to prevent the trigger altogether.
So you're not done yet.
No.
You're not resting on your laurels.
No, 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 absolutely not. I think, also if you have someone in your life that's impacted, and I don't, once you start on this journey, you just like want to do as much as you can towards moving therapies, or, maybe I don't have the best, but this will be the proof of concept to the next best, to the next best. So I think, you always have to keep pushing, and that's what I hope to do.
Well, we really appreciate the work that you've done, and we're very excited about what's to come. Dr. Sheikh, this has been an excellent conversation. I think you've given a lot to think about to our patients and family members who are listening today. Send some sunshine up north, and I understand you're coming home back to see family in Toronto soon. So very happy holidays to you, and thank you so much for spending the time with us today.
Thank you so much. Again, I am eternally grateful to you as patient advocates to, get the message out to the community, the other patients. Thank you for all of your efforts in this very, very severe disease. But, we are at the forefront now. So I think we have the tools.
Exciting times. Thank you.
Thank you.